This is Anatomy Physiology, Chapter 11, where we're going to look at the muscle tissue, but we're going to look at it at the molecular level all the way up to the tissue level. And there are three types of muscular tissue, and they are the skeletal system. And we talked about skeletal muscles in Chapter 10. In this chapter, we're going to talk about the cardiac, which would be your heart muscle, and then smooth muscle is also known as your involuntary. Your subconscious takes care of it. You don't have to consciously tell the smooth muscle to contract. So what are the things that you can do with your muscles? Well, one thing that muscles and nerves have in common is that they are excitable, and they actually have an electrical charge across the membrane. So we're going to spend quite some time talking about that. If you learn it well in this chapter, when we get in the next chapter and start talking about nerves, we'll just be saying the same thing. Instead of saying muscle cells, we'll be saying neurons. Instead of depolarization of a nerve membrane, in this chapter we talk about the depolarization of a muscle membrane. In both nerves and muscles, the electrical depolarization or the conductivity travels the length of the muscle fiber or the length of the axon in the case of a nerve. But muscles are different because they contract. That's what happens to them when the electricity runs through them, which is different than what happens with a nerve. And you're able to also stretch the muscles. And again, nerves pretty much don't do that. We'll start out with the three different types of muscle tissue. With the skeletal muscle, it is voluntary, which means if you don't talk to it, if you don't send it some sort of message through a hormone or through a nerve impulse, it'll just sit there. It won't do anything. They are usually attached to bones, but in the case of the erector pili, that one is connected to a hair. The hyoid bone isn't attached to another bone. We learned about that. But it does have some muscles attached to it. I mentioned the hyoid because it has at least 8 muscles that attach to it above and below, but it itself is able to move. So that's kind of strange. It floats. Usually we think about a muscle being attached to something, and it has to be firmly attached on the origin, and then the insertion point is the part that moves. But in this one, both the origin and the insertion can move. Another place you have skeletal muscle that is not attached to a bone is in the face. So a lot of the things that you do when you're frowning and smiling use muscles, but they're not attached to the bones. So how would I know that I'm looking at skeletal muscle? Well, it's striped. So that kind of gives it away. They call it striations. And this is what it looks like under the microscope. So clearly, stripes. We'll find out in just a minute that those bands are actually from thin and thick filaments. So where they're thinner, it looks lighter, and when they're thicker, it looks darker. They also can slide across each other. So we'll learn about how they contract. It says they're voluntary, under your conscious control, but they can also be stimulated with hormones. So that's another way. But we'll talk more about that when we do the endocrine system. Now, interestingly enough, muscle cells are made up of fiber, and they can be a foot long. So that's pretty long. 30 centimeters is quite long for a cell. But because they are so tiny, you still need a microscope to be able to see them. Here's a couple of pictures I found using the electron microscope. Associated with these muscle fibers, we also have nerves, which will stimulate them, and you have blood vessels. Last chapter we talked about Endomysium, Perimysium, and Epimysium. So here's your Endomysium, and it's wrapped around these muscle fibers. So this one is encased in a muscle fiber. You need to be able to bring this around and make this into a tube. So this is a slice through, so you can see inside. And this is actually a real picture too. It's not a cartoon picture. So you can see a little bit of the nucleus. Here's the nucleus. There's the nucleus. But this isn't just a tiny band. It is in circles. Here's a look instead of from the side where you've sliced into it and you're looking at the individual muscle fibers. Here we are looking at the end. So here's a muscle fiber, here's a muscle fiber, here's a muscle fiber. Here's another picture. These are each muscle fibers. And then that white connective tissue surrounding it, that's the endomysium. There's the endomysium. And then you have a whole bundle of these. So here's a muscle fiber, all of these are muscle fibers. And then if you look, they're held together there by the perimysium. And then if we go down here, we can see that here is a bundle with the perimysium, here's a bundle. And then over it, you have the epimysium. Epi is upon, para is around, so it goes around, and endo means inside, so there's the endomysium. And then you have the tendon, and you have the fascia over this whole thing. These connective tissues are made of collagen, and collagen is usually thought of as not flexible, but it is a little bit, just slightly. The more collagen you have, the less flexible it is. So we talked about collagen in dense, regular connective tissue and dense, irregular connective tissue. I really got a kick out of this picture because those connective tissue bands are holding your muscles in place, and one of the things that you notice when you're working with your abdominals is that you get six-pack abs, and here we have two gentlemen who have gotten six-pack abs simply by leaning up against a fence instead of having the connective tissue hold the muscle into nice six-pack definition. If you're talking about the cell membrane of a regular cell, we call it a plasmalemma. And if you're talking about the cell membrane around a muscle fiber, we call it the sarcolemma. Sarco means muscle. And then if you are thinking ahead to the next chapter, it's going to be called a neurolemma because it is the cell membrane around a neuron. So they just changed the beginning prefix to indicate what kind of cell the cell membrane is surrounding. Instead of calling it cytoplasm, which is what you would do in a regular cell, we call it sarcoplasm. And myofibrils are long protein cords occupying most of the sarcoplasm. So once you get past the cell membrane and you get into the actual muscle fiber itself, you're going to find that there's not much in there. There's, of course, a lot of mitochondria because you need a lot of energy. But you're going to find mostly it's just these long protein cords. They're called myofibrils. Two other things that are a little unusual that you find in muscle cells is glycogen. So when you have too much sugar in your bloodstream, you pull the sugar out and you put it in your liver and you put it in your muscles, and your muscles convert it to glycogen, so it's a kind of a short-term storage form. So that when you're out running or doing whatever it is, you don't have to stop and eat. You can just start breaking down the glycogen that you've got stored up in your muscle fibers. There's also another pigment that we're going to talk about. It's called myoglobin. Anytime you see healthy muscle, it is a bright red. And this is in part from the blood that's in it, but also there's a pigment called myoglobin. And it has a higher affinity for oxygen than hemoglobin does. So your muscles are going to snatch a lot more oxygen out of the bloodstream. And you need a lot of oxygen in order for the muscles to work. One of the things that disturbs me is the insistence that they not call muscle cells. They call them muscle fibers. But most of us, when we think of a fiber, we just think of it of a little protein rather than an entire cell with all of the organelles and things that you have to have inside of a cell. So I find it extremely confusing that they insist on calling muscle cells muscle fibers. But they call them fibers because they're long. Like I said, they can be up to a foot long. Another thing that's interesting about muscle fibers and a little bit disquieting is that you can have multiple nuclei. So when you're going from being a child to an adult, you are stretching and adding more protein in your muscles. And as you are tearing the plasmalemma, the sarcolemma, then you are fusing your muscle cells. Now, we know from the chapter on cells that if you have multiple nuclei in a cell, the cell doesn't know which one to listen to. So it's frequently a cancer cell. And your body sends in the macrophages and your immune system to destroy the cell because it has more than one nucleus. But in the case of the muscle cells, they don't divide. So they get bigger, but they don't divide. So they don't usually become cancers. So it is okay to have multiple nuclei in a muscle cell. When we were learning the parts of the cell, we learned about the endoplasmic reticulum, and we learned that it is kind of a conduit. It's a system of tubes that transport stuff around inside the cell. And now when we're looking at the muscle fiber, which is really a muscle cell, we're going to call the endoplasmic reticulum the sarcoplasmic reticulum. And this is going to be surrounding the myofibril, which actually is a fiber, the long fiber running through the cell. And it is important because it stores calcium. So in order for your muscles to work, you have to have calcium. And you need to have it sometimes, because you don't want your muscles working all the time. Sometimes you want it available and sometimes you don't want it available. So you can put your calcium into the endoplasmic reticulum, which is now called Sarcoplasmic Reticulum, and then you can release it as you need it when you need the muscles to contract. Spend some time memorizing this picture. This is going to be extremely important. If you don't understand this, you pretty much aren't going to understand the rest of this chapter. So just if you have to stop the video and look at it and think about it, or if I'm not making it perfectly clear to you and you don't understand it, go look at a YouTube video of somebody else explaining it. But make sure before you go on past these next couple of slides that you know exactly what's going on. So let's start up here. This is a muscle fiber, which is another name for a muscle cell. And if you notice, it has multiple nuclei. Then if you go past the cell membrane, which is now called the sarcolemma, but that's the cell membrane, the outer phospholipid bilayer. Inside, you're going to see mitochondria, and quite a few mitochondria. And then each of these things inside the cell is called a myofibril. So don't get the muscle fiber, which is the whole cell, confused with the myofibril, which are these long proteins that run the length of the cell. And there's quite a number of them within each of your muscle cells. Now this bottom picture is where they've taken just one of the myofibrils out of the muscle fiber, out of the cell, and we're looking just at the myofibril because this is the thing that's going to cause the cell to contract. So that's why it's so important to understand what's going on here. So they've taken just this section right here of one of these myofibrils, and this is what it looks like. So surrounding it, you've got the endoplasmic reticulum. Now, again, we're not going to call it endoplasmic reticulum. We're going to call it sarcoplasmic reticulum because it's surrounding a muscle. And this is your reservoir for the calcium, among other things that it does. And if you look at this, you have a light area and a darker area, and then a light area, and then a darker area, and then a light area. And so what you're actually looking at is what makes the striations. So this is why we call it striated skeletal muscle. Another thing that's confusing is the tendency for different lab manuals, different textbooks, different editors to define the Z-line or Z-disc or Z-body. Your textbook calls it a Z-disc. So here's one Z-disc, and you come over here to the next Z-disc, and that makes up a sarcomere. Sarcomere. And then if you went from here, this Z-disc, to the next Z-disc right here, then there would be another sarcomere. So there's a series of sarcomeres running the length of the myofibril. So most of the books that I have looked at call it a Z-line. It makes a line across there. Sometimes I like to throw a little trivia in. So here's something I found that was kind of interesting. In the Z-disc, also known as disc with a K, Z-line, Z-band, this is where the actin molecules are going to attach, and we're about to learn what actin and myosin do. But this is the interesting factoid. The Z-disc is composed of hundreds of different proteins, and it is one of the most complex macromolecular structures in biology. So it'd be kind of interesting to know what all kind of cool stuff is going on within the Z-disc that allows our muscles to contract. The area on either side of the Z-disc is called the I-band. And then in between, where you have the dark area, this is called the A-band. And then here's another Z-disc with its I-band. And then we start another A-band. So we're going like that. And then right in the middle, we have the H zone right there. And this is the M line. Got all that? A sarcomere, the repeating unit that runs down the myofibril, goes from Z disc to Z disc. And within the Z disc, you have the I band, the A band, the H zone, and the M line. Here we have the blow up, and then we're going to then blow it up even more and show you exactly what's going on. So these dark lines right here are actually myosin, or some people call them the heavy band, and then actin is the light band or the smaller one. So in this particular picture, they've kind of made the actin green right there, and they've made the myosin purple. And then this M line right here and right here and right here, those are where the heavy bands of myosin are, right in the middle. And they call that the H zone, where the M lines are. So that's where you have your heavier bands. And if you look, they interdigitate. Here is actin, here's myosin, here's actin, here's myosin, here's actin. So they overlap on this end and on the other end. So that's what this blow up is showing you here. Here's the myosin and here's actin on either side of it. And now again, remember this is a flat view, so bring that on around. They're trying to show you here that this is circular. But when you look at it like this, you think it's just flat like a piece of paper, but it's not. So bring some more actin around here, and some more actin around here. Some more here, some more here, and behind it. So it's surrounded in a circle by the actin. All right, you got that? All right. If I were the boss of the world and I got to change things, one of the things that I would change is I would not call this the A band, because that would make you think that that was actin. I would call that the M band for myosin. But I'm not the boss of the world. Somebody else decided to call it the A band, and we're kind of stuck with it. But don't let that confuse you. The thick bands right there are myosin, and the thin ones are actin. And the actin is attached to the Z disc. And on this end, these are attached to the Z disc. So now that they've taken it apart for you and showed you the individual pieces of it, now they're trying to put it all back together for you so that you can kind of see. So here's a muscle cell, which we're going to call a muscle fiber. And there's its cell membrane, which we're not going to call a cell membrane. We're going to call it the the sarcolemma. And then you get inside, just inside, there's a nucleus, here's a nucleus, here's a nucleus. Right there. So it has multiple nuclei. And then these are your myofibrils, not to be confused with the muscle fiber. And each of those has sarcoplasmic reticulum around it, which in other cells would be called endoplasmic reticulum. And remember, they are a reservoir for calcium. And then if we strip away the sarcoplasmic reticulum, then we were able to see there is the myosin band, there's the actin going this way, actin going that way. So you know where the actin meets right there is called the Z disc or the Z line or the Z band, depending on which book you're reading. From here to here, those are all the A band. So it's the length of the myosin. right there. All right, so hopefully this is coming a little bit easier for you as we go over it a couple of times. And then in between these myofibrils, you have lots and lots and lots and lots of mitochondria because you're going to be needing a lot of energy to make these muscle fibers contract, relax, and then contract again. Oh, and then let's throw one more word in. So we have the muscle fiber, which contains myofibrils. And within the myofibrils, we have myofilaments. And those myofilaments, of course, are going to be actin and myosin. Now we're going to drill down and we're going to actually look at the myosin itself and we're going to look at the actin itself. So we're getting down to the nitty-gritty. We're getting right down to the molecular, the chemical composition and seeing what's going on. So I think that the myosin's kind of neat. It looks like a golf club. It's got the shaft and it's got the head on it that you would be hitting the ball with. But we're going to use that head of the golf club to ratchet the actin and cause the muscle to contract. So let's see how that's going to work. Here's one myosin molecule, and then there's the head of it. So there's the golf club looking piece. And now we're going to have a bundle of these, and there are all the little heads sticking up, and they're all around. So this is nice. You can see it's a 3D effect. And that's what makes up your thick filament. That's what makes up your A band. In the middle, that's where your M line is, and that's where your H zone is. Right there, in the middle of the myosin. So here's the actin sticking out in there. And then there's the myosin with its heads. And then this is the bare region. Another one of the myofilaments is the actin, and it's going to be intertwined strands. And going along the strand, there are active sites where the head of the golf club of the myosin can fit. So that's going to be really important. Now, I don't want my muscles always contracting. I just want the ones that I want to contract. I need some for muscle tone. But if I'm just sitting around, I don't need my muscles twitching. So what I'm going to do is I have another molecule called tropomyosin, and it's a rope-looking molecule that wraps around the actin and covers up the active sites. So the myosin head can try and attach to the actin, but the tropomyosin gets in the way. It blocks it. It's like taping it off, so you can't get to it. And then along the tropomyosin, there are little troponin molecules, and they hold on to each of the tropomyosin molecules, and they're activated by calcium. So remember the Sarcoplasmic Reticulum where we have that calcium? So your myofilaments are actin and myosin. And then you have tropomyosin, which wraps up and covers the active sites on the actin. And then you have the troponin, which looks a lot like tropomyosin, so don't get them confused. And it's this small calcium binding protein on each of the tropomyosin molecules. So the red represents the actin in this particular model, and it's a double band. So it looks like somebody's taken a string of pearls and twisted them together. And then overlaying them is the tropomyosin. And then holding them together is the troponin. So it's almost kind of like they're thumbtacked onto there. And now we're going to add one more myofilament. So I'm going to show it to you here, and then we'll go and read about it. So here is your actin, that's red. And here's your myosin, which is purple. And if you look, connecting to one end of the myosin and the other end of the myosin is at what looks like a green spring. And I think it's fun that the name of it is Titan, like you're tightening something up, but you spell it Titin, Titan. Now look where it goes. So here's the myosin, and so it's being held to the Z disc on either end. So it's suspended. Here's another one. But look what goes on the other side of the Z disc is the actin. So here's the actin. There's the titin. There's the myosin. And there's the titin. There's the Z disc. And there's the actin again. So they kind of alternate. So titin is huge. It is springy. It runs through the core of the thin filament, which would be your actin, and it anchors it to the Z disc and to the M line, and it stabilizes and holds the myosin in place. And it also prevents overstretching, and it allows for recoil. So it's kind of that's why it looks kind of a springy coil. So let's see if we can recap and put it all together. So here's your I band right here on either side of the Z disc. Here's another I band. Here's your A band, which is where the myosin is. Here's your M line, and that's kind of easy to remember because it's in the middle. It's the M line. And then this band right here is the H band. Right there. And we're lacking those golf club heads. So here's an actual picture of all this stuff we've been talking about. So here you can see the bands right there. Right there. There's the actin, which makes up the I band. Here's the myosin. Now, let's see what happens. That's your muscle contracting. If you look carefully, you'll see that these bands go away. Let me back that up a little bit and let you watch it again. So look at the light and the dark band as it contracts. You see them going away? So here it is, spelling it out for you. The tropomyosin, remember, is covering up the active sites on the actin, and the troponin is like a little thumbtack that's holding the tropomyosin in place. As soon as you release calcium and it combines with the troponin will actually change shape because the calcium is now on it. And as it changes shapes, it'll pull the tropomyosin off of the active sites on the actin. So now the individual heads can touch the actin, and then with the aid of ATP, you're going to flex the head of the golf club, and you're going to ratchet the actin and the myosin past each other. So that's why I wanted you to see that video, so you can actually see the movement of the actin and myosin as they slide inward towards that middle line, and then release, and then slide forward again, and then release. Here's a picture of someone's hand who doesn't have calcium. So for some reason they're not dissolving their bones fast enough to keep enough calcium in their bloodstream, or they're not eating enough calcium, but for whatever reason it isn't in their bloodstream, and so the muscles can't access it. They can't store it up in high enough concentration in the sarcoplasmic reticulum. And so you end up with your hand frozen or your feet frozen in these weird contorted positions. They are spasmodically flexed. So it's like you had a spasm where it flexed and then it locked. Tetany means locked, not moving. We've been looking at just one of the myofilaments. We've been looking just at one of the sarcomeres on one of the myofilaments. But if you take the whole myofilament, then you've got the actin, the myosin, all those stripes that you see. And you have something called dystrophin. And why this is important is if you don't have correct dystrophin, you can't cause the muscles to work correctly. So we have a condition we call muscular dystrophy. People with muscular dystrophy end up in wheelchairs because their muscles don't work the way that they want them to. This is what normal muscle fibers look like, the endomysium. And this is what someone who has muscular dystrophy, this is what their muscle fibers look like. So under the microscope you can see that it is not at all normal looking. So let's start right here. That should look familiar to you. You should see that and go, oh, that's a phospholipid bilayer. That must be the cell membrane. And indeed it is. It's the sarcolemma, which is the cell membrane for a muscle cell. And then outside you have the endomysium around there. It's the connective tissue that kind of holds the whole thing together. And then you have a series of proteins that are connecting the endomysium through the cell membrane, through the sarcolemma, and on the inside of the muscle fiber or the muscle cell, you have the dystrophin, which is attached to the actin. So it's anchoring it in place. So this is going to help you with your muscle contractions. And this is why people with muscular dystrophy who have this thing impaired are unable to make their muscles do what they want them to do. And this is a summary in words. It gives the where the word comes from for the A bands and the I bands. I don't care about that. It's just the way the light passes through the bands. This is an actual picture of a sarcomere, and it's not a cartoon picture. They've actually taken this with through a microscope. And look at the size of the nucleus compared to a sarcomere. The sarcomere goes from this Z line to this Z line, this Z line to this Z line, right there. And the nucleus is way larger, way larger. So it's nice to put those in perspective. And this picture does a nice job of that. One of the things that's kind of interesting, as those actin and myosin interdigitate, slide across each other, the Z lines are pulled closer together, but neither the actin or the myosin changes length. Only the amount of overlapping changes. In the very first chapter, we talked about how atoms go together to make up molecules, and molecules go together to make up macromolecules. The macromolecules go together to make up organelles, and organelles go together to make up cells. Cells go together to make up tissues. Tissues go together to make up organs, and then organs go together to make up organ systems. And then organ systems make up an organism. So I don't know if you remember back then. So what they're trying to do here is to kind of give you the hierarchy from most complex to least complex. So we're starting out with a muscle. So here's our muscle, and we have the tendon, and we have the epimysium, and then we have the perimysium, and then if we look closer, then we have the endomysium. Within the fascicle, we have muscle fibers, which again is a name for muscle cells. And then within the muscle fibers, we have myofibrils. So there's your myofibrils and your myofilaments within the myofibrils. And then each myofibril can be broken up into a succession of sarcomeres, sarcomere, all the way down there. And then if you look at the sarcomere, which goes from Z-line or Z-disc to Z-disc, then within that you have the myofilaments, not to be confused with the myofibrils, myofilaments. And the myofilaments are actin and myosin and tropomyosin, troponin, dystrophin. So by now, I hope you guys have gone over this, and you can name all the little parts, and you can do the hierarchy from most complex to least complex, and you're not getting tangled up with myofibrils and muscle fibers and all of those words that seem so close together, troponin and tropomyosin. So spend a little time, and then we'll go on. So now we're going to talk about how does a nerve interact with the muscle. How does it talk to the muscle and tell the muscle to have the actin and the myosin slide across each other? So we're going to need nerves to talk to the muscles and cause them to contract. Although you can do it with hormones also. There are chemicals that you can cause the muscles to contract. But generally, especially skeletal muscles, want to have a nerve stimulate. And you control it. So we consider skeletal muscles voluntary. So your brain sends a message saying, do this. Or we're going to learn about reflex arcs where you don't need the brain involved. If the nerve is not able to talk to the skeletal muscle, then the muscle will actually shrink. So they call it atrophy because there is no nerve talking to it. I remember a number of years ago when my daughter had to wear a cast on her leg, and when they took the cast off of her leg, because she hadn't been using that leg, she had been using the other leg, but the one that had the cast on it, she had used crutches or something to keep her foot off the ground so she wouldn't put any pressure on her leg. And when the cast came off, one of her legs was normal size because she'd been using it, but the one that had the cast on it, the gastrocnemius muscle was shriveled up. And so she had one little chicken leg and she had one normal curvy calf leg. And it freaked her out so badly. I remember that. And I just said, well, you just need to keep working your leg and you'll get your calf back. And she did. So you can not grow new muscles. If they had died because of the cast, then she couldn't get them back. But because they just atrophied from lack of use, she was able to then exercise and get her calf back. So now she looks normal. Doesn't act normal, looks normal. As her mother, I'm allowed to say that. All right, somatic means body. Motor neurons, so these are things that cause your body to move. And nerve cells that are in the brain stem and the spinal cord are going to come out and talk to the skeletal muscles. When we get into the next chapter on nerves, we're going to talk about that a nerve has a cell body. So that's the main part of it, where it's got its nucleus and all of its organelles and things. But then it has processes sticking out from it. And the branched processes that stick out on one side of it are called dendrites. And then the long, long process that leaves the cell body and comes on out is called the axon. And we're going to learn that dendrites bring information into the cell, and the axon carries the information away from the cell. So if you can remember, axons away. So we're going to talk about that in the next chapter, but if you can hang on to the fact that the axon is the business end of a nerve, then you'll be able to follow through this next set of information that we're going to go through. So axons will go to the skeletal muscle, and at the end, you're going to branch out and touch a number of muscle fibers, or remember, muscle fibers are actually muscle cells. And here's an important point to remember. Each muscle fiber, or each muscle cell, is supplied by only one motor neuron. So you'd short circuit it if you tried to have more than one motor neuron telling that muscle cell what to do. A couple of chapters from now, we're going to be talking about the spinal cord, and this is a slice through the spinal cord, and here's some nerves coming off, and they are innervating, they are talking to the muscle fibers. So here's a couple of them. You've probably heard about fine motor control and gross or coarse motor control. So you have these muscle cells, and it says on average there's about 200 muscle cells. If you have a small bunch of motor units, then you're going to have a fine degree of control. So this is where you're going to have your eye, because we need quite a bit of control over our eye to be able to see, and then our hand muscles to be able to do the things that we can do with our hands. But other places, you're going to have large motor units, and what you're interested there is just strength rather than control. So you just need to be able to lift or move things. So you have larger motor units with hundreds of these muscle cells. So just two of the really big muscles that you have, your gastrocnemius, which is your calf muscle, and your quadriceps femoris. Remember, the femur is the largest bone in your body, so you've got a really large muscle controlling it, or one of the many large muscles controlling it. So it can have up to 1,000 muscle cells for each neuron. Now, once upon a time, we called nerve cells, but then somebody pointed out that a bundle of axons is called a nerve, and they said, oh, well, then we can't call nerve cells anymore, so we're going to have to rename them neurons, so that when we say the word nerve, people know that we're just talking about a bundle of axons, and when we say the word neuron, we're talking about a nerve cell. So for each 1,000 muscle cells, there's one nerve cell that's branched off and talking to it, to those muscle fibers. A synapse is the place where a nerve fiber approaches its target cell. It approaches that muscle fiber, that muscle cell. And I am changing the word meets to approaches because it never actually gets there. So the nerve can't touch the muscle cell or the muscle fiber because it'll short it out. So it stops just short, and so a synapse is actually a gap between where the end of the axon came, branched out, and it gets close to the cell, close enough to boss it around and tell it what to do, but not to touch it because they're both electrically charged. So you don't want them touching. Kind of weird. I added this word to the slide because for some reason it's left off, but it's one of my favorite words, and most of my students enjoy it too. It is a button, but you pronounce it bouton. And you got to get it up in your nose, bouton. So this is a cartoon representation of the bouton and how it interacts with the muscle cell. So up here is a nerve, and coming off of it is an axon, and the axon comes down and it branches off into these axon terminals or boutons, and this broadens out. That's why they call it a button, a bouton. And inside of it are little vesicles, little bags of acetylcholine. So there are other neurotransmitters, and we'll talk more about the other neurotransmitters when we get into other chapters. But for this, since we're talking mostly skeletal muscles at this point in time, you're going to have these boutons, which all of these bags of acetylcholine are, which is a neurotransmitter. And when an electric event occurs, a depolarization occurs, then you're going to release the acetylcholine into this gap, this synapse. So notice this does not touch. You have to have a gap. So you don't want to short this out with the electricity in this going and having the electricity in this one. So we're going to release this chemical, the acetylcholine, into this synapse, and on the muscle cell itself, on the cell membrane, which we're going to call the sarcolemma, we have these little receptors for acetylcholine. So isn't that handy? Here we have acetylcholine here in the presynaptic membrane. And then this is the postsynaptic, or the other side of the synapse, and it's got the receptors. So this is going to come across, it's going to touch the receptors, and it's going to cause a reaction inside the muscle. And notice the mitochondria hanging out, ready to supply ATP energy, because this is going to take a lot of ATP energy. So this is called the presynaptic, because it is before you get into the synapse, and this is the postsynaptic. So the presynaptic is the axon itself. It's the bouton of the axon, and the postsynaptic is the actual muscle cell, which you're going to call a muscle fiber. And right here we have a series of sarcomeres. Here's a sarcomere, there's your actin, there's your myosin. And so I'm going to be a spoiler alert and tell you that once you get the acetylcholine across the synapse and you activate the acetylcholine receptors, you're going to get these muscle fibers to contract. And here's 4 different pictures I found. I think this one is my absolute favorite because it shows exactly what they were saying in words before. Here is a muscle cell or muscle fiber. Here's one, here's one, here's one. And here is a neuromuscular junction. That's where the neuron and the muscle come together. And each of these little things are boutons. So here's your axon coming in and branching off into these little buttons. And remember, each of these little buttons is full of bags of acetylcholine. And they're going to release the acetylcholine and cause this muscle to contract. This one is my next favorite because you can actually see the bouton, and you can see the light and dark bands. You can see the striations. So that's a neuromuscular junction. And this is a bunch of neuromuscular junctions. And here's 3 more pictures. These are at much, much higher magnification. So here you can see the synaptic cleft. This is the synapse. And you can tell which is the muscle and which is the nerve, because remember, the nerve's got all those little bags of acetylcholine. So you can see all of these bags. So you know that this is the presynaptic, or the bouton of the axon right there. And then over here, you've got the muscle cell itself, which would be the postsynaptic membrane right there. And so there's actually a picture of a synaptic cleft. And to keep these two from touching, you've actually got some proteins in there kind of holding it apart. So it's kind of like anti-Velcro. Here's an even closer picture of it right here. Here are your bags of acetylcholine, your vesicles. Here's your synaptic cleft right there. And then there's your muscle fiber, which is your muscle cell. And so the acetylcholine is going to come out, go through this cleft, and stick onto the receptors on this side. And then this is a scanning electron micrograph picture. So here comes the axon coming in, and you can see it branching off. And it doesn't, I wish they would come on a little further and show you the bouton out there, but it's just showing you how it's coming in. Like this picture. It just shows you it laying across, but it didn't go all the way to the end where the bouton lived. It's hard to decide whether to show you the picture first and then give you all the words, or give you all the words first and then go back and show you the picture. But anyway, I wanted to show you the picture first for that one, because look at all these words. There's a lot of them. All right, so the synaptic vesicles, so those are those bags of acetylcholine that are in the bouton of the axon. They undergo exocytosis, so they fuse with the membrane, the end of the axon, and release the acetylcholine into the gap between the nerve ending and the muscle cell. And then the muscle cell, which is a muscle fiber, has millions of acetylcholine receptors. So they attach the acetylcholine. And in the second half of this lecture, when we do the second half of chapter 11, we're going to talk about myasthenia gravis. And people who have this condition they don't have enough receptors. So where I said you have millions of acetylcholine receptors, these people do not. And so it's going to cause severe weakness. They're not going to be able to contract their muscles as thoroughly as they need to. When we get into the next chapter on nerves, we're going to learn about Schwann cells, named after a guy named Schwann. And we're going to find out that it is an insulator for a nerve cell. But we're not into nerve cells yet, so we're just going to skip over that part, and we'll pick it up next chapter when we know what the heck we're talking about. All right, now you've got the acetylcholine that jumped across the synapse, and it caused the muscle cells to fire. But now I need my muscle cell to quit firing. I need that to stop. So you have acetylcholinesterase, which is, when you see the word ACE, it means it's an enzyme. And so this is the enzyme that's going to break down acetylcholine so it can't act on the muscle anymore. So now the muscle's going to stop firing. The calcium's going to go back into the sarcoplasmic reticulum. And I'm going to just sit and wait until I get another message to excite the muscle and cause it to contract again. So you got acetylcholine jumping into the synaptic cleft, which starts the whole muscle contracting. And then to stop it, you've got to remove the calcium and you've got to break up the acetylcholinesterase. Now, there are other neurotransmitters that I mentioned besides acetylcholine, and some of those are also degraded by an enzyme. They're broken down and then reassembled. And others, they are just reabsorbed back up into the nerve ending. So we'll talk a little bit more about that again in the nerve chapter. It's hard to separate the two chapters. When we come back and start talking about the next part of the chapter, we're going to talk about watching the whole muscle contract, not just the individual sarcomeres. We're going to look at the whole muscle. And so here is one of the things that they used to do in labs, and I haven't seen them do them in labs in a while, but we used to take a frog and we would cut its leg muscle out, and then we would attach it so it is suspended here and suspended there. And then you put an electrical stimulus to it, and you can cause it to contract. So this is showing you that it really is electricity that causes a muscle to contract. So here's the electricity coming past these two, and every time he touches it, you see the leg jump. And then attached to the bottom of the muscle is a pin. So the tracing that is being made right there shows us each time the muscle contracts. And we're going to find out why it's suddenly plateaued like that in our next episode.